Hi, how are you?
It's okay. Yes.
Thank you. Thank
Thank
you
you
very
for
much.
inviting me.
Thanks for joining us today. We are looking forward to your talk. So you published a recent paper in Nano Letters. Exciting work.
Thank you.
So before we go to your seminar, maybe you can briefly introduce yourself and then start sharing your screen. Thank you very much.
Of course. So my name is Diana Sobota, and I work at the University of Cambridge in the Cavendish Laboratory and the group of Professor Ulrich Kaiser. And our group has a very strong team working with lipids, and they make really remarkable lab-on-a-chip devices where they form model membranes and run all sorts of tests on them. But I look at lipids from a slightly different perspective, which I will be talking about now.
Thank you.
So, are you ready? Can I?
Yeah. Just start sharing your screen and we're up.
Okay. Can you see the presentation now?
I can see in the presenter view.
Okay. Let me try it again. It's very annoying. Just give me a moment.
I mean, if it's presenter view, we can also live with it. It's fine.
Okay, I think I will manage to sort it out now.
Yep, looks good.
Okay,
Yeah.
perfect. Yes, so the paper that I will be talking about, and also I will tell you slightly more from behind the scenes, is about tailoring interlipid lipid transport. And as I mentioned, I look at lipids from a slightly different perspective than my colleagues. In particular, I'm really fascinated by the level of control that lipids themselves have over organism and cell. Most of the time when people think about cellular membrane, they think that it's the proteins that do all the work. And lipids are more like a gray mass that just provides a structural scaffold, and that's it, which is a very common but still a misconception, because when you think about it, lipids and lipid bilayer have a very precisely controlled composition, and especially the asymmetry that I will be talking about. So it's so precise that it cannot be just coincidence, and in fact, it is not. It is very important what lipid species are on which side of the membrane, on the inner or outer leaflet, because each lipid species can act as a signaling molecule. And in nature, there are proteins that can flip them from one side to the other and trigger some specific mechanisms. And just to give you an idea how important it is, it's, for example, one of the signaling mechanisms for an apoptosis. So we're literally talking life and death. And we looked at it and we thought, yes, okay, it has huge importance in a cell. Think how amazing it would be to build a structure. So go to the lab, open your fridge, and there is a whole library of different structures that can control your membrane in that way. And this is basically the starting point of what we were doing. And you see, we have, if we just simplify a membrane with these two components, we have lipid membrane and something that will control where the lipids are. We have these two materials, and I will just briefly tell you how these materials look like. First, the gray mass, which is our lipids, the very important part. In the cell, well, cell as a construct is extremely complex. For a physicist, I I would not really want to describe that as a physicist. So how about we simplify it and just look at the membrane, only cellular membrane. However, the membrane itself is already really, really complicated. Like all of the proteins that sit there and will mess with our experiments, we don't really, we don't need that. So how about simplify it more and look only at the lipids? And this is what we did. So we'll be looking at just spheres made of lipids, which are called lipids. And in this case, again, it is even more simple. We'll be using just a single component membranes, which are zwitterionic POPC. If you remember the composition, POPC, PC lipids are on both sides. There is an abundance of them in the cell. And this is how our spheres look like. So we have plenty of them. They are in the cellular size range. So perfect. We have something to work with. And now for our synthetic construct. The most important part of this material needs to ensure precise molecular structure, because that's exactly what proteins do. They had many, many years to evolve to have a certain shape. Well, we don't have that time, but we have something that nature provided us with, which is DNA, the molecule that we can very, very precisely control, well, in terms of shape. So you know DNA as this double helix that appears everywhere. And you probably know that the way this helix is formed is not random. In fact, it's very specific the way the two strands are combined. We need to stop thinking about it as this double helix and unwind it into two separate strands. And now these strands, we can buy them. They're commercially available. We can design them very precisely so that they match exactly the way we want. So for example, in nature, we have two long strands that come together and they work like a zipper. Very, very specific in terms of binding. Now, there is something that we're missing here. It's a very important chemical detail that we will need later on. DNA is very strongly negatively charged. So it's not that simple. Two very strongly negatively charged molecules will not come that close together to bind. So we need to add them some positive ions to act like bridges, then they can nicely come together and form these zippers. Now let's make it a little bit more interesting. We can have plenty of these little strands, design them in a way that they can form, for example, something like this. This is already pretty simple, but you can see that we have an amazing amount of control over structure at the nanometer scale. This is something that I don't think any other material gives us, except for proteins, but it's really difficult to design them to have the shape that you want. So now we have this design. It's very simple. You see that the scale, the dimensions of it are roughly similar to the ones for transmembrane proteins that we will try to mimic. The only problem is we can buy this, we can design it, buy the strands, fold it. It will take you maybe a day, and you have plenty of these. The problem is, if we want to put them in a membrane, they are absolutely useless. And that's because the strong charge would prevent them from going into the membrane. So then we need to use another property of DNA that is its huge advantage, namely how easy it is to chemically modify it. We can place, I think, really any chemical modifications that we want with the resolution of 0.3 nanometers in exactly the place we want. So how about we put something that will work like an anchor? And if we design it properly, the beauty of it is that it just fits in a membrane. It fits exactly the way we want it. And it may not be as complex yet as the proteins, but you can see that it's a great prototype, that we're already have an idea for a scaffold, our structure. And so before I start really explaining the results and what we did, I know that the audience here is mostly, I think, people that are fascinated with lipids. And you will notice that in this work, the lipids are more like a medium that we're using. It's very simple, right? Just one component and just a lipid vesicle. I mostly look at it from the point of the structure of the construct and how to design it with the thought of a lipid in my mind. But for you today, I think I would like to propose that you treat this whole talk as an advertisement for a material that could work as a probe for your membranes. So I would talk about just one specific story that we come up with, but think of the whole process of what we did, and I think you'll see that there is a huge potential of this material and how we can design it to study plenty of other features of membranes. I think maybe return to this later, but have this in mind, and here we go. So first of all, what I wanted to make sure we understand is what actually happens there. Because DNA, even though it looks similar to proteins now, it's a great prototype in terms of shape, it has very different chemistry, right? So how does it actually look like, the pore and the surroundings? Well, as I mentioned, DNA is very strongly negatively charged, which makes it very strongly hydrophilic. Now, membranes formed of lipids that will have hydrophilic group in their head. So we'll get something like this. I think it's pretty logical, right? We put DNA in there, we form a pore, and now the lipid tails will not go anywhere near the DNA. They really dislike it. So all the head groups will now form something that we call a toroidal pore. We have hydrophilic head groups facing the hydrophilic surface of DNA. Now in the pore, that gives us a hydrophilic environment. Means there will be water there probably. We'll also talk about this. Now my friend was really fascinated by this, and together with the Alexei Aximentiev group from University of Illinois, they actually designed a structure very similar to this, put this in a membrane, and Alexei simulated the results. And they learned that indeed the toroidal pore will form. We'll try maybe. So in this case, they decided to color the lipids from one leaflet pink, from the other one green. You can see if the highlighted lipid, for example, the green one, will slowly move here to the pink part of the membrane. This is because this is the most amazing part of this, and we'll base our whole idea on the fact that these lipids now can move from one leaflet to the other. Initially, the two leaflets are separated, right? So lipids from one side cannot move to the other one. That's what also provides us with this very specifically controlled asymmetry. But right now, with the toroidal pore, we have two leaflets merged, and lipid from outer leaflet can very easily, just via simple diffusion, move to the inner one. And you can see this with the highlighted lipids. So what they did now is we can now take a closer look at the pore itself. And if we color code the leaflet again, well, with different colors, but I'm sure you get the idea. Remember the asymmetry. This will be the composition of the outer leaflet. This will be the composition of the inner leaflet. After we form the pore, we very quickly lose this asymmetry, which means we can change the asymmetry, or more precisely, we can just shuffle lipids randomly. How about controlling this change? We do something to really understand and, well, to really make sure we know what the lipids are doing. And for this, we thought, what is the reason this happens? The reason is DNA is hydrophilic, or rather the membrane spanning part, this part is hydrophilic, which in turn makes this pore form. The hypothesis that emerged from this was the hydrophilic DNA will form this toroidal pore. But if we manage to put something hydrophobic in the membrane spanning part, there is no reason for the head groups to be in the pore. There's no reason for them to merge, which means perhaps we'll just be, we'll end up with a cylindrical pore. And this is the basic idea behind our design. We're kind of going back to the basics. So earlier you saw there was a big construct, but now let's just go back to our very simple zipper, just the double helix of DNA. We first need to make sure that DNA goes into the membrane. And this is why we have these two anchors, which are in this case cholesterol molecules. Now we have our membrane spanning site. What will we put there? Well, first of all, we can just have DNA there. So it will be very similar to what I showed you before. We expect a beautiful toroidal pore. Now, how about one side of it will be modified internally with a dodecane, which is carbon chain of, well, a chain of 12 carbons, very strongly hydrophobic. So this will be something like this. Of course, I'm sure you know we don't expect something exactly like this. We'll get back to this. And of course, we can also place two dodecane spacers there. And now we have three structures, two dodecanes. I will use these names, so remember that. Two dodecanes, one dodecane, zero dodecanes, which is just pure DNA. And they vary in terms of the hydrophobicity of the membrane spanning site. I will just one last time make sure that we know that each of these constructs will be modified with cholesterol. So there are two different hydrophobic modifications. Each of them has cholesterol that ensures the insertion, make sure the DNA is inside. And the other one will be dodecane, which is now something that we hypothesize will control the shape of our pore. And these are the sketches that we'll be using as well. Okay. So as I meant, something else that I think is really important. I like to think about this as, let's imagine this whole story, what happens there. We have our vesicles, we have just a pure membrane, we add this particular DNA. What happens is it needs to find the membrane first, and then it inserts, which means that before it reaches this orientation, it needs to puncture through the membrane with this part. This is like a rod of very strong hydrophilicity. So for some time during the insertion, DNA will be in the membrane. And if DNA is inside the membrane, there will be a toroidal pore formed, even just for a brief moment. If we have toroidal pore, so hydrophilic surfaces together, we have hydrophilic environment in the pore. So we have water. So together, again, with Alec Aximantiev from Illinois, we looked at it with O-atom MD simulations. Our three structures, we looked at the water channels inside the pore. And you'll see that the more hydrophobic the membrane spanning site is, the more water we get. There were lipids there as well, but we removed them just for the clarity of the image. So we have a lot of water surrounding both DNA strands, and now, especially here, it's very well visible that there is no water around the dodecane molecule, while a lot of water molecules will surround the DNA. And here, the two dodecane spacers do not have water facing it. You may wonder why there is water here in the channel at all. This is because when we think about it, if we would form a pore that looks exactly like this one, the cylindrical one, we will not have any means of testing it. Because we don't optically see this DNA, we look at either water, ions that flow through it, or lipids. So this construct is designed with the dodecane being slightly shorter and shorter than the span of the hydrophobic core of the membrane. So there will still be some water in the channel. If we have water in the channel, we'll probably have some ions. And this we tested experimentally with a voltage clamp. It's basically just a chamber separated with a membrane, When this membrane is intact, there are no ions flowing from one side to the other because membrane is permeable. So if we measure the current flowing through the membrane, we of course get nothing. There are no ions flowing, the conductance of the membrane is 0. However, if we put now DNA through it, and DNA spans the membrane, there will be hydrophilic environment, water, ions, we'll see very sudden step and a lot of ions flowing. So what we would expect is all these structures inserting and forming, when they insert, forming these steps. What we get is this. So you may think that, wow, it looks really noisy, but it is not. What it shows is that these constructs, they change a lot. So they don't just insert and stay there. They constantly span the membrane, go in and out. So these are transient ion channels, these DNA structures. And this is also the way they were designed. I won't go into the details, but if you have questions about it, I will be very happy to answer this. Also, so that we can see better what happens with our DNA, especially with the case of 2D construct, that if it would form a cylindrical pore, we would not see it. So here we want to see the differences between them, not necessarily completely close the channel for the 2D. So let's go to what is really interesting for us, which is the lipids. How will we test what happens with the lipids? We already know that there are differences between these three structures. We can see it with water, we can see it with ions. What will they do with lipids? We need some means of testing it. And in fact, there is one that is used in biology for people who work with natural scramblases, which is called the dithionide assay. Dithionide is the molecule, as you can see here, very strongly negatively charged. Remember that, it will be pretty important. It has two features that are interesting for us. First, it is a very strong reductant. So if we have fluorescent label that is sensitive to the reductant, we can put dithionide there, and from this fluorescent molecule, we'll get a bleached one that does not show any fluorescence. In this case, it's NBD. It's very easy to also get NBD labeled lipids. We'll use that. So we have NBD labeled lipids. We add dithionide, we lose the fluorescence. The other important part is dithionide, also because of its strong charge, is membrane impermeable. So if we now have our lipid vesicle with 2 leaflets, both of them labeled with NBD, we add dithionide, it will not bleach the inner leaflet because it has no way of getting there. And of course, lipids do not just on their own spontaneously move to the other leaflet, which means in this case, we'll observe something like roughly 50% loss of fluorescence. And in this case, as I mentioned, we are using POPC. I added 0.5% of NBDPC. We don't need all of it to be made of NBDPC. We just want to have our probes. Then we added the dithionide. Only outer leaflet is bleached, 50%. What happens if we now have a pore? So in this case, well, I drew you the pore, but you can imagine that this pore was formed for a reason, and in our case, this reason will be DNA. If we have that, and we now add dithionide, first only the outer leaflet will be bleached, but because of what I mentioned, the diffusion of lipids from one side to the other, all of it will be bleached. All the lipids will move now. All the lipids from the inner side can very slowly, through diffusion, move to the outer leaflet and be exposed to dithionide. So the whole membrane will be bleached. So in our case, our goal is to see if we can form this pore with the DNA and if we can see this bleaching and what will be the differences between three structures. I think at this point, you're probably thinking, okay, Diana, there is a huge hole in the membrane. And this dithionide, it's not really that big of a molecule. So can dithionide leak through the spore? And maybe it would just bleach the inner leaflet because it leaks. Well, the answer, of course, is no. Well, we tested it. And you probably know why negatively charged molecules will not go easily next to the DNA. So DNA pore is a natural cation selective pore. And this is again from Alexander's paper, where he performed this control. And if you take a look at our recent paper in Nano Letters, this is very well explained, and also we talk a lot about why negative ions will not go through the membrane via DNA pore. Okay, so I think we have this. I think you're ready to take a look at the actual assay. So we expect something like this. We have NBD labeled vesicle, we add DNA inserts. If we add dithianide, the lipids from the inner leaflet will flip through the pore, and we see all of it being bleached. So the final intensity of the fluorescence will be 0 for our vesicles. There will be no fluorescence. In fact, we see something like this. So these are histograms of final intensity of the fluorescence of our vesicles. And some of them indeed go to 0, which means they have all the lipids flipped. But you will see that there is also another peak, quite huge actually, of vesicles that don't seem to have anything happening to them. So these vesicles have their intensity bleached to 50%, which would suggest that there is no pore. And I mentioned this so strongly because I'm very much fascinated with why this happens. And I will be very happy to discuss this, especially with people who know probably much more about lipids than me. But I will not focus on this. I have some hypotheses which I can share with you later. Let's now forget about this and only think about the ones that have lipids flipped. So these are the ones, the vesicles where we know our DNA was working the way we designed it. Now, our free structures, we look at the time traces of how the fluorescence was changing, and the most important results of this experiment is, depending on the hydrophobicity of the membrane spanning site, we get different rates of flipping. And we can also see this by eye, basically, that the structure that is very hydrophilic, the one that has no hydrophobic spacers whatsoever, it flips lipids very quickly, go, again, relatively quickly, and goes to 0. The other ones also go to 0, again, because the cylindrical pore is not entirely closed because of the design, but the hydrophobic spanning site makes it more difficult for the lipids to move through the pore. It's not so easy now. They don't have a clear pathway. So it is visibly slower, even though they end up being of the same intensity, well, 0, because these are the average vesicles that showed whole bleach, 100% bleaching. And if we this time get rid of one of the cholesterols, so remember the 2 cholesterols were there to ensure the insertion. If we now have just 1 cholesterol, the insertion will not happen. So this is basically our control, the structure that attaches but does not form a pore. And these vesicles, none of it ever showed 100% bleaching. So one of our conclusions will be, if we change the membrane spanning site, we can change the rate of transport. Now, let's take it forward and think about what happens if we start changing something else in the environment. The important part, I already mentioned this, but this is very, very important. So once again, positive ions are important for the way this whole system behaves. In fact, I would say ions are one of the most crucial part of controlling what happens in the membrane in this system. And because of that, it's very common that whenever one designs a structure and works with DNA, we have a set of very standard characterization methods, and we usually test our structures in different concentrations of ions. So I did that, and I won't go into the details how these methods work. The most common ones are gel electrophoresis, and we're measuring the melting temperature. The only thing I want you to notice is the two structures that are modified, so the turquoise and orange one, which is 1D and 2D, behave very differently than the one that does not have a modification. So the yield of folding of these structures is much lower in lower amount of magnesium. So if we reduce number of cations in the system, we get lower yield of structures. And similar here, the stability of these structures, when we change magnesium from 4 millimolar to 1 millimolar, changes a lot in contrast with this structure that does not have such big change. So these are the only two important parts that you need to remember, that the stability of the internally modified construct is affected by the magnesium concentration. And you may think maybe it's because they are less bound, because they have less smaller number of nucleotides, which is like the segments that form the but you see that I also performed a control that shows us that even the unbound structure does not show such big difference. It must be something about the internal modification that makes them being less stable. Okay, so I know that probably for people that deal a lot with lipids and not with DNA at all, this may be confusing, but let's just remember this one part. Internally modified constructs are sensitive to changes in magnesium. This made us think, okay, so how about we perform exactly the same thing? So we're still aiming to flip lipids, but what if we now change the concentration of magnesium during the assay? So initially everything is fine, DNA attaches, inserts, and the lipids are diffusing. But now when we add dithionide, we also dilute the concentration of magnesium. So now it is much lower at the end. And also it's decreasing during the experiment. Let's look at our histograms. They're really interesting. We learned that the structure that is not modified behaves roughly the same. We'll talk about the differences in a moment, but it has two peaks, again, by model distribution. Some of them did not do anything, some of them flipped. Okay. The interesting part is 1D and 2D constructs, so the ones that have the internal modifications, do not show complete bleaching anymore. So we have no vesicles that would bleach completely. And we hypothesized that this is because their stability was affected. So they never managed to flip all of the lipids during the assay. So, but they already started. You see the peaks are not just on 0.5. It's not that they didn't do anything. It seems like some of the lipids were flipped, but some of the internal inner leaflet lipids stayed inside and were not exposed to dithionide. So we managed to stop flipping during the assay. And if we again look at the time traces, they look roughly like this. We average over the vesicles measured, which starts resembling how proteins look like, because they can stop whenever they want. So of course, we're nowhere near the level of control that proteins have, but this is already very promising. And we decided that this combination of our DNA construct, lipids, and magnesium is very promising. So let's look at it more. And you can see already something that I didn't mention, but let's talk about this now, that 0D, so the unmodified construct that is supposedly stable with the magnesium concentration differences, also does not behave exactly the same as it was when magnesium concentration was constant. Why is that? obviously that was our question. And what other mechanism is playing a role in this whole system? So let's look only at 0D from now on. And let's think of how ions affect this. So we're going back to gel electrophoresis that I did not explain yet, but I will now. The intensity of these bands, simply put, are the yield of folding, which means in this case, this construct is folded in 100%. You can maybe see that this band is slightly less intense, which means when there is literally no ions, it has huge troubles coming in together. But these two strands are very are so long that there is such a strong force bringing them together and wanting them to be together, that even though there are no cations, these two negatively charged molecules will overcome the repulsion and come together. I'm showing you this to make sure that we know that in case of this structure, the stability is not of any issue. Even though we change the magnesium concentration, the construct is always folded, maybe slightly less here, but it is still there. However, when we put it on lipids, on lipid vesicles, and try to see what it does, because of it, with the fluorescent label, we can now see what the structure does. Surprisingly, we see that it does not attach, even though it has the hydrophobic anchors, even though it is folded properly. If there are no cations, it will not go near the membrane. Then we can start adding maybe more magnesium. We slowly start seeing something, and more. We see that the more magnesium we add, the more constructs bind to the membrane. If we plot the peaks of these histograms on versus the concentration of magnesium, we get this beautiful hill curve, which shows us how, with changing concentration of magnesium, we can control the number of attached constructs. And remember, these constructs are also functional, which means with the amount of magnesium, we can so precisely, just using this curve, tune how many structures will be in the membrane and how functional, and basically the level of their activity in the membrane. Then we thought of why this happens, right? Because we know that it's not the stability that is affected. So perhaps it's something about the very charge of DNA. So when you look at this, there are 48 base pairs, which means there is 96 monomers of DNA, each of them having a negative charge. We have 96 charges, and 2 hydrophobic anchors that are supposed to drag this whole rod of negative charge into the membrane. What if we make it a little bit more difficult for it and only put 1 anchor? So we have only 1 cholesterol. As you probably remember, it will not insert, but it will still attach to the membrane. Let's try it. Try to perform the same experiment, and then we see that the curve changes, which means right now, with the same number of charges, but smaller amount of hydrophobic anchors, it is more and more difficult for it to drag DNA to the membrane. It needs more magnesium, more cations to ensure the same attachment. We can also try to go the other side and make a very small structure with single cholesterol, like this, where we learn that, in fact, if there is very few charges per the anchor, it is very easy now. We just need a little bit of cations, and we ensure that. So we concluded from this that in this case, it's the screening on the DNA that is of the highest importance. You may think that it is a disadvantage, but when you look at this, we realize that this provides us with a beautiful switch for our structure. We can now decide exactly how many structures are attached. And by that, you can remember how many of them are functional in the membrane. So now let's try to perform an experiment that shows if it's reversible, if we can really control it. And we'll use a molecule called EDTA. It's the way it works, it chelates divalent cations. So if we have just DNA with vesicles with no DNA and no magnesium, there is no attachment, right? Like you remember here, nothing attaches. If we add magnesium, we see the attachment. Then if we add EDTA, we hypothesize that EDTA will capture the divalent cations and take them away. So we perhaps see loss of the attachment, and this indeed was the case. So it was a very fun experiment to perform, just sitting there and adding consecutively magnesium, EDTA, one by one, and seeing the rings appearing and disappearing one by one. This works, but now, well, obviously, we don't really care for the little fluorescent rings to appear and disappear. We want to do something with it. So if you remember the ditanite essay that I told you, and if you think about the fact that for the functionality, the attachment is crucial, we can use the exactly same idea to show that we can, with the magnesium, we can now switch the lipid transfer on and off. Well, in this case, only on. And the experiment looked very similar to the initial one, only in this case, DNA is not attached. So there is no magnesium. The DNA still has cholesterol. It's very important. It is possible for it to insert and flip lipids, but it doesn't because there's no magnesium. If we add ditanite, then there are no pores. So we only observe 50% loss of fluorescence. If we then add magnesium, DNA attaches, inserts, flips lipids. Lipids are exposed to ditanite. The whole vesicle is bleached. So these are the results. Single frames from the experiment. They match with the numbers here. So we have initial 100% fluorescence and no attachment. Then the loss to 50%, still no attachment, but then when it attaches, it further bleaches the vesicle. And you can, I think, better see it on the trace, where we can now trace our NBD lipids. With 100% fluorescent, they go down. You see the little ruffle, that's where I added magnesium, goes down, but also DNA ring, this one, starts appearing, reaching its 100%. And after some time, we see this additional drop, which means we managed to switch on the lipid transfer. So with cations, we can control the attachment of the structures, and with the attachment, we can control their functionality. Yes, so to sum it up, with these particular DNA constructs, and remember, this is just one single story. With these constructs, we managed to flip lipids. We managed to do it at all, which is remarkable. But also, we managed to do it, well, to control the rate. Obviously, it's not control that that the proteins have, but it already shows you that with just a little bit more designing and careful designing of the experiment and of the construct, we may be very close to creating a single structure that can flip more and less depending on certain stimuli. What's more, we learned how they respond to cations in two ways. One, the very first one, with the internal modifications that had their stability affected. So with the stability, we managed to stop the flipping at a certain point. But also, we learned that screening of them is so important that we can switch the flipping on and off using cations only, which is, by the way, I think I mentioned this already, but the really cool thing about this is that this is exactly what proteins do. The actual molecules that were designed by nature to flip. They use divalent cations to switch on and off their flipping. Okay, so going back to our very first idea of building a synthetic structure that can control the membrane. So, I mean, you already know that the answer to this is we're not there yet, right? I mean, if we manage to form a structure that has the level of control that proteins have, you will hear about it probably from a Nobel lecture. But in this case, we're trying to show that there is something inspiring about these structures, and we can work on making them act as beautiful probes for your lipid membranes. This is, I think, the most interesting part for me. We can induce changes and we can start controlling them. And there are so many possibilities of doing this. Thank you. And with that, I will just thank the funding bodies that made this possible. Also, my group, especially Ulrich, our principal investigator, and Alex, who did the foundations under this work. Also, the Michele group and Aximantive group, who were very remarkable staff of Lipids. Thank you.
Thank you very much, Diana. These are fantastic tools. So of course, we'll have some questions. I think I will start with my own question first. So when you show this DNA structure, right? So you have the cholesterol in the middle, cholesterol on the other chain, and then basically you still have hydrophilic end of the DNA here and here. But this hydrophilic end still, it goes through the membrane.
Yes.
How can it go through this hydrophobic core of the membrane by itself?
Okay, this is a very good question. This is, okay. I will just share my screen and try to, no, I can't do it now, sorry. Okay, I will just use my, I will just explain it by talking. So, you remember the bimodal distribution on the histograms? Let's think of what would be the difference between vesicles, because we looked at all the macroscopic details of it, like size or initial intensity, and there seem to be no real reason for this to be. So this shows you that 50% of the huge membranes do not have any DNA inserted. And I think this is very closely related to your question, because the insertion efficiency of this construct is in fact really low. But just not to point out the disadvantages of my construct, the reason I believe this happens is the construct has cholesterol on the terminal end of one structure. Okay. Screen again. Can you see it now? Can you
Yeah,
see
yeah, I do.
So this is the sorry. Okay. So this is the construct, and you see there is cholesterol here. There are in fact 4 strands. And there is a break here.
Mm-hmm.
And if we imagine that this DNA attaches to the membrane with one cholesterol, the idea is the other cholesterol pulls it in, because it cannot twist, it cannot flip. This is very stiff, by the way. The upstream DNA is really, really stiff. So the DNA lies on the membrane, inserts with one cholesterol, and then the other cholesterol pulls it in. However, I believe that with these constructs, the vesicles that did not show any insertions are the ones that have had DNA with these bases broken and the cholesterol twisted and inserts, which means the DNA just lies on the membrane. And as you meant, as well, this is where your question came from. DNA will not go there without being really forced to. So the way we deal with this right now is we basically close this gap. If we close the gap, we we don't give this the flexibility to turn, and this now ensures 100% efficiency. This is not published yet, so you don't see it in this paper, but I'm really glad that you asked this question. This is something that I'm really fascinated about.
Okay, so basically, you know that this would be the case. This is the reason. This is actually good that you know it. And do you see any dependence of the size of the vesicle, depending on?
Yes. Yeah. This was the first thing I looked at. I was thinking perhaps is the size, but no, there is none, and I really don't see any. However, I believe that in some way there must be a difference between different curvatures, but for these structures, they're so small that with giant vesicles, they basically see the membrane as flat. I think with small vesicles that would have also nanometer diameters, there would be a difference.
Okay, so our first question is by Fred Heberle. He says, great talk, thank you. And then, do you know roughly how many pores there are per vesicle under conditions where you see rapid flip-flop?
I think this is a very good question. And it's because initially we always assume that, again, I'm going back to our histograms. When you look at them, you would think that statistically, it is reasonable to assume that these constructs have 0 pores, so these have just 1. Because it would be very unlikely to say these have 100 and these have 0 and we see nothing in between, right? So initially we thought, okay, so how about this one just have 1 pore and this have 0? But right now, I don't think it's the case. I'm sorry, I know that you probably wanted a number, but no, we don't have that. And in fact, seeing the insertion efficiency would be a great advantage to us. But the only thing I can say is I don't believe it's just 1. I think it is more, but because of the transient behavior of these pores, we simply can't really tell.
But when you fluorescently label your construct, you see a lot of it, no?
Yes, I will go back to this. So this is what we see. These have, let's say, 10 micrometer diameter. DNA has, well, I would say 12 nanometer in length. And you really can't see any separation. So I don't think we can count the number of fluorophores. There is just, well, but remember, these are attached constructs. So there is a huge layer of DNA attached because they really want to because of the cholesterol. But I think it's very difficult for them to really flip through the membrane and being inserted. We can't really test how many of them is.
True. Okay, so next question is by Milka Doktorova, and she's asking, how do you add mixed DNA with the vesicles? Can you try different protocols for the addition and incubation, for instance, temperature, stirring, to help the uniform distribution across the vesicles?
Very good question. So actually, when I started thinking about this, I was always thinking about molecular biologists who they have plenty of protocols where they have cells, well, and their membranes, add DNA that is not modified with anything, that they want to push through to prepare some, to act as vectors for transfection and so on. So they do it in 3 ways to help this insertion via temperature changes, electric pulses, and with surfactants. So yes, we try a lot of different protocols. In this case, DNA is simply added there without mixing at all, but we just leave it for an hour at least and wait for it to diffuse, so that kind of not to really disturb them. However, this is a very good question because it's really interesting to try different ways and try to see how they affect the membrane. So for example, we can change the temperature, make it a little bit more fluid, and then perhaps the DNA will find it easier to insert. Similarly, a very common thing to do is add a surfactant. It also messes up with the membrane a little, makes it easier for it to insert.
And following this question, did you try different compositions and see if they could react differently?
So I think when you're asking this, you mean some compositions that are really complex, like actual cells. We didn't.
Not actual cells, but let's say a bit less rigid, more rigid.
Yes, we did. And it's also a very, very exciting part of the work. We're trying to see how different phases affect this, because the other side of the story with cations, which I did not mention because we're still in the process of thinking about it, is depending on the phase, even the same lipids, they have their head groups arranged slightly differently. And with beta ionic lipids and ions, and with different head group concentration, different head group orientation, you have completely different electrostatic interactions with the membranes. So we did not look at the mechanical properties of the membranes as such, but we looked at different phases and different compositions in terms of, for example, adding a little bit more cholesterol to make it a little bit more ordered, and see how this affects it. And in fact, there are huge differences.
So you get less, probably.
Well, most importantly, if the gel-phase vesicles, the gel-phase membranes, because of the way they electrostatically interact with ions, surprisingly, they do not need these anchors, the cholesterol anchors, to get the attachment. So in this case, well, okay, they also need magnesium, of course, because it will now be based not on hydrophobicity, but electrostatic interactions. But with no cholesterol, but a lot of magnesium, we'll still see attachment, which is not the case with gel-phase lipids.
Okay, so Fred asks again, I wonder if one could estimate the number of the pores in the membrane using simple assumptions about the size of the pore, size of the vesicle, and rate of the lipid diffusion through the pore.
So what we did, for example, in our group we did for the paper that Alex wrote, the very first one that showed the flipping, we had a very simple assumption that there is a single pore, and with that they managed to calculate the rate of flipping. Well, yes, calculate the rate of flipping, and it agreed very well with the simulations. However, I'm not sure how I would do it without assuming the number of pores, but actually looking for the number of pores. I think especially because they are transient, so there's one inserts and goes out, and they can swap places. It would be very difficult, but actually I would love to just give it a try. It must be probably not that difficult. The question would be how reliable that is. Well, I would much rather have a proper assay that will allow us to really test the insertion efficiency and see them really recently or whatever. Yeah.
Thank you, Diana. So one more question. So now you basically incubate, and the questions are always about the incorporation of this construct to the membrane. So for let's say cell membrane, do you think they will also incorporate with the cell membrane with passive incubation, or you should do something else?
So Alex, in his paper, this first one, had some cell work, and he showed that they do induce flipping with cells. So
But
yeah,
for this, probably one pore will not be enough, right? But you should have a lot of pores so that you would see the effect.
I think so. That's why I think the most important part would be to have a model membrane, do something simple, and come up with some protocol that will allow it to very reliably insert and maybe sit there stably. And from my side, I tried it with certain cells, and we do see not only incorporation, like attachment to the membrane, but surprisingly, with many cells, they spontaneously go inside. And actually, it's not something that I know that much about, but I believe that with the, for example, with the concentration of magnesium, that we have to ensure bigger constructs are folded properly, and with certain environment, the cells are just unhappy, maybe. And so again, I think one step before working with cells is to make sure that we can fold the DNA in the conditions that cells would find comfortable.
Okay, thank you very much. I think we had all the questions covered. Thanks a lot, Diana. That's really fantastic tool. I'm sure a lot of people will make use of it. Thanks for sharing this with us.
I hope that because it was so focused on not on lipids, you didn't find it
not at all, not at all, not at all. So the audience is not only lipid focused people, they're also a lot of in general membrane biophysics people. So I'm sure they'll find it very interesting.
Great.
Okay,
well,
thank you.
It was really a pleasure.
Thank you very much for your time.
Thank you everyone. Thanks everyone for watching. Bye.
Bye.